My name is David H. Nguyen. Thank you for attending this seminar. Today I'm going to talk about methods for quantifying hidden spatial information in tumor architecture. The algorithms that I'll be discussing have automated R scripts that can be downloaded from my GitHub page, and you will see the link in the following slides. In this presentation, I'm going to briefly talk about recurring patterns in tumor architecture, and then I'm going to explain 3 of the algorithms that I've developed and how they work, and give specific applications of them. We know from histopathology that the alignment of cells within tumors is non-random. Otherwise, it would be impossible for a pathologist to say that this tumor is an adenocarcinoma, or that this tumor is a squamous carcinoma, or that this one is a spindle carcinoma. The fact that we can classify recurring patterns into specific histological subtypes means that the arrangement of cells within a tumor are non-random. However, beyond gross morphological features such as ones before you, it is difficult for the pathologist, the human eye, to pick out subtle patterns of order inside a tumor. Thus, it is difficult for pathologists to subclassify each of these categories into distinct subtypes that may have different response to therapy or different prognostic outcomes. From gene expression studies, we know that a tumor will express the genes into proteins too that are reminiscent of their tissue of origin. For example, here these 3 tumors are mouse mammary gland tumors, and if the tumor came from, say, the outer basal layer of the mammary duct, its gene expression profile will be reminiscent of the basal layer cells of the normal duct, and its morphological features will form these stretched out sheets that are often appear as streams of cells, as you can see in panel F. If a tumor came from the luminal layer, that is the inner layer of the mammary tube, then its gene expression program will be reminiscent of the luminal cells, and its morphological features will form these bricks stacked side by side. Thus, there is plenty of evidence that the tumor architecture is not a random process, though it is what we call highly heterogeneous process, because it's seemingly random to us, because we do not know how to assess the information or make sense of the information that is there. And thus is the premise of my research program in developing algorithms to quantify this heterogeneity in the form of spatial information. So the question that I ask is, instead of resigning to this term heterogeneity, that is, it's too complicated for us to make sense of, I ask, how can we objectively quantify spatial information amid disorderly patterns? And I try to answer this question through computational algorithms. The first algorithm that I'll talk about today is called the Quadrant Slope Index, or QSI algorithm. The purpose of this algorithm is to quantify cellular alignment patterns within a disordered tumor architecture, for us to further subclassify a tumor beyond its general histopathological designation. The human eye is easily distracted by disorder, which is why something like camouflage actually works in hiding people in the forest. If you look at panel A, here's a cartoon of a tumor. It'll be obvious to the human eye that in this upper right-hand corner, there's these dark pink cells that are lined in the relatively same row, and that their slopes point to the upper right. And here, this little group here, their slopes point to the bottom right. However, most people will miss that in this bottom left-hand corner of panel A, there are also many nuclei that are lined together, that is, in a row, pointing towards the bottom right. And if you were to divide this tumor into individual units and count up all the slopes that point up and down, what we can reveal, as in panel B, is that there are hidden patches of ordered nuclei. By ordered, I mean aligned, their slopes aligned in the same direction, right? And so the human eye can pick out certain things very well. Here, the green squares that are obvious in panel A. However, there's a lot of hidden information here in the red squares that the human eye has a hard time assessing, especially if you're looking at a tumor that can be very large on the scale of tissues. So the quadrant slope index method, what it does is that it takes an image of a tumor and it divides it into quadrants, in this case 4 quadrants. And in each of the quadrants, what we do is we measure the longest length of each nuclei, and then we measure the slope of the angle that straight line makes. And here is the reference. Assuming the x-axis is horizontal, then any line pointing to the upper right is a positive slope, and any line pointing to the bottom left is a negative slope. And so in this way, for each of these quadrants, we can count the number of green slopes going up and the number of red slopes going down. And if we divide green over red, what we get is a number. Numbers that are larger than 1 means that there are more green slopes than there are red slopes in that square. And numbers that are less than 1 means that there are more red slopes going downward than there are green slopes going upward. So since this method turns each square into a number, and this number can then be translated into a color. In this case, red and green. If a box has the same number, if a square has the same number of red and green arrows, then it is the color yellow, which is what you get when you mix green and red. But if there are more green slopes in a box, then it will seem more green. And then if there are fewer, or there are more red slopes in a box, then it'll appear more red. In this way, we can translate the spatial information in a tumor into color pixels. And here's an example of what that does. So this is a biopsy of a human breast cancer. And this is its one representation through the QSI algorithm. And as you can see, it's really hard to pick out information, spatial information in the hematoxin eosin image of this tumor, because there's just too many things going on. However, if we were to divide this up into individual grids, in this case a 12 by 12 grid, and then count the number of slopes in each square going up and going down, what we find is that there is spatial information, describing the morphology of this part of the tumor. That is, if you look at the outer edge of the tumor, a lot of the slopes are downward, actually. And that if you look at this inner edge, there are some patches where a lot of slopes are pointing upward, right? And then in between are oranges, where there's a transition zone between the upward and the downward pointing nuclei, right? In this case, we only mark the epithelial nuclei, not the stromal nuclei here in this fibrous region, which is this blank gray area. And so we can see from here that there's a lot of spatial information that can be extracted from an image, and that things like borders are very important, because we know from studies that the extracellular matrix at the border of a tumor is much stiffer than it is in the inside of that tumor, right? And that stiffness dramatically changes the biological properties of the cells, right, in that region. Thus, the quadrant slope index method can extract really useful information from routine images that are just too complicated for the human eye to assess. So beyond visualizing orders of regions of order and disorder as defined by slope alignment, the QSI algorithm allows us to classify large samples numerically without looking at them. That is, we can classify tumors by the percent of squares that are reddish or greenish. And here I say reddish or greenish because these colors are thresholds, right? We can define a cutoff value above which we define a pixel or a square as red, right? Or below which a pixel is green. And there's all sorts of ways that we can classify. That is, through ratio of green to red, and so on and so forth. And so this method, by turning spatial information into numericized information, we can apply rigorous statistics to our samples. The next algorithm I'm going to talk about is called the linearized compressed polar coordinates algorithm, or LCPC. The purpose of this algorithm is to objectively quantify complex shapes. In biology, we often encounter odd shapes such as this amoeba here, and rarely do we encounter nice, neat shapes such as this ellipse, or this circle, or this diamond, or this square. And so it's very difficult to objectively quantify the information in this shape of this amoeboid structure, because we can't say it's circular or it's squarish, right, or it's ellipsoid. It just has an amoeboid shape, amorphous shape. And thus, we need a method to capture this complex spatial information. And I did this by applying a radial grid system. In this case, the inspiration for developing this algorithm, the LCPC algorithm, was to characterize colon polyps, benign colon polyps. And so here is a cartoon of a colon polyp. Okay, it is a bulge out of the colon wall, and it forms a lot of tubes that are disordered inside of it compared to the orderly wave tubey structure of a normal colon lining. By applying this grid of 0 to 180 degrees, right, we divide this image into individual sectors represented by these radial lines. And if every time we marked, okay, if we wanted to measure the tubiness of this polyp, then we mark every time one of the radial lines crosses the border of a tube. And we do this for every line. And then we can sum up all of the points for each line such that every line only has 1 point. And here in panel C, I've color-coded red, blue, and green to correspond with panel B, which lines I'm talking about. So this in panel C is called the LCPC transform of the spatial information in panel A. And the neat thing about the LCPC is that it is a discrete sinusoid wave. Okay, it's a complex wave that has discrete points. And such a wave can be transformed by the fast Fourier transform into a handful of frequencies. So in this way, the LCPC algorithm turns a very complex spatial shape, like the tubiness of this polyp, into a handful of numbers, that is in the form of frequencies, right? Frequencies that comprise the sinusoid waveform. So if we were to apply the LCPC transform to the basic shapes we started with, we see that each shape has a distinct LCPC signature, frequency signature. A circle has a frequency of only 1, and that makes a lot of mathematical sense in terms of how we measure frequencies, that is the time it takes to do 1 revolution. An ellipse has a distinct frequency signature. So does a square and a diamond. Okay? But most exciting is that even very complex amoeboid shapes can have distinct frequency signatures. In this way, we can classify shapes by their objective numerical frequency signatures. If we were to apply this to actual case, here is on the left side, panel A is a normal colon lining, and on the left is a sessile serrated polyp, that is a colon polyp that forms tubes that have these sort of jagged teeth patterns. And if you apply the LCPC algorithm to the normal colon, what you get here in the middle is its LCPC profile, and then you can apply the fast Fourier transform to derive the frequency signature that represents the tubiness of the normal colon. You can do the same for the sessile serrated polyp on the right. You can do the LCPC transform to get the discrete sinusoid wave, and then you can apply the fast Fourier transform to this wave to get a handful of frequencies that represent the spatial information in these jagged toothed polyp tubes. And so in this way, the LCPC transform can objectively quantify shapes, complex shapes in tissues, in the form of numerical frequencies, in the form of numbers. And this, again, allows us to apply rigorous statistics in classifying colonic morphology based on numerical objective features. As another example here, this is a pedunculated colon polyp. That is the polyp that bulges out from the colon lining. And here, if you can apply the same LCPC approach, except in this case, the origin is 0, 0. And in the previous cases, the origin I chose to be just y equals 0. And then, so all the radial lines emanate from the same horizontal line. Why that's the case, I will not discuss here. It is discussed at length in the manuscript. So I recommend you take a look at that if you want a detailed discussion. But for now, I just want to show that you can apply the LCPC in a radial manner, capture the spatial information of the tubiness, all the tubes inside this polyp, in the form of this sinusoid wave, and then you can do the fast Fourier transform on this wave to get a frequency signature representing this shape and the arrangement of tubes within it. And I want to point out that this LCPC algorithm can be applied to any type of image, really, not just histology, but CT and MR data. So, but that will be in a future presentation. The third algorithm I'm going to talk about is called the numericized histogram score, or NHS algorithm. The purpose of this algorithm is to quantify and visualize preferential aggregations within heterogeneity. So here, if you look at panel A, here is a distribution of 4 types of dots. You have black, blue, green, and red dots. And to the human eye, we can pick out that the blue dots tend to be near the black dots and that the red dots tend to be near the green dots. And yet these 4 categories are separated from each other by this space in the middle. But if you were to change the color scheme from solid colors into these distinct checkered patterns, what you get is image in panel B. The image in panel B is the same as panel A, except we switched the solid colors for these recurring checkered patterns. And it's obvious that it's much more difficult to pick out which categories are hanging out together and which are far apart based on these checkered patterns, because again, the human eye is easily distracted by high levels of disorder. But this is the very challenge that a pathologist face. If you look at panel C, here is an H&E image of a breast tumor. And you can see that there are distinct nuclei based on their chromatin patterns, right? And so the pathologist can see that, yeah, there are recurring types of chromatin patterns that appear in this tumor image, but it's very hard for the pathologist to see and keep track of who is likes to aggregate with whom. Thus, I developed the numericized histogram score to solve this problem. Assuming that we can define which category every object belongs to, in this case you only have 4, black, blue, green, and red, what you can do is that for every point, you can measure its distance to its closest neighbors. In this case, let's focus on black number 17, which is this item here. In panel B, I measure the distance of black 17, right, to every single blue dot in this system. In panel C, I do this something similar for black 17, but now I measure its distance from every red dot in this system. And then in panel D, you do the same for the black number 17, but now you measure the distance of this black 17 to every single green dot in the system. In this way, you can see that if you do that for every single black dot, not just black 17, what you get is a distribution of distances. In this case, in panel A, this is the distance of black dots, all black dots, right, to other black dots. And panel B is the distance of all black dots to all green dots. And then so on and so forth. For C and D is the distance of black to blue in C, and then black to red in D. Now, if I didn't show you the panel on the left, and I only showed you these histograms, and I explained to you how I derive these histograms, you would be able to derive that these black dots tend to be near other black dots. As you can see, that most of the bars are in the bins on the left of the dotted line, and that the same for blue dots. Black dots tend to be near the blue dots because the distance between black and blues fall within these smaller bin distances, right, the left of the dotted line. So if you look at panel B, you can tell that some green dots are close to black dots, while many green dots are far from black dots. And the same for panel D. Some red dots are close to black dots, but then there are also many red dots that are far from the black dots. And these distributions make sense of what we know the system to be. And so the advantage of measuring these distances in the form of histograms is that the numericized histogram score can turn this distribution, any distribution, into a number, a single number. Here's the equation. I will not go into details because it's in the manuscript. But essentially, the novelty is that you add a weight, positional weight, to every bin in your histogram, and you multiply the number of items in each bin to its weight, so you get a product. In this case, you have 10 products because there are 10 bins. If you sum the 10 products, you get a number, and you divide that number by what I call the maximum saturation here, and you get still another number. And so the advantage of the NHS algorithm is that it turns histograms into single numbers. So here's an example. Here is a number of distributions, histograms. And if you look at the top and the bottom, these are mirror images of each other. And the ones just inside of it, these two, they're also mirror image of each other. And the blue to gray, that's one system. That's the NHS algorithm to generate that number, that one number. Each histogram has a S score. That is, this one has a score of 0.85, and the one down here that's opposite of it has a score of 0.25. And on the column on the right, this is just a transformation to use two colors. And if you want your S score scale to straddle 0 and be larger, as opposed to just being between 1 and 0. And so these methods, the NX score allows you to capture the information in a spatial distribution in the form of a histogram into a single number. And these numbers can then be translated into a color coding system. The ultimate advantage is what? Is that you can visualize, excuse me, the aggregation of different categories of things with each other. For example, here in panel A, it shows the dark purple ones are the black dots that are close to other black dots. And thus it makes sense out here that these black dots are gray because they're far from all the other black dots. And then if you look at, in panel B is the black dots to green dots. So all of these squares are black dots. And the fact that they're light purple and gray means that they're far from green dots. While the black dots on the right side of panel B are dark purple because they're what? They're close to green, many green dots. And the same can be described for C, black to blue, and D, black to red. So in this way, the NHS algorithm can extract proximity information of 4 categories that are very hard for the human eye to pick out. But if we can define which category each dot belongs to, then we can run the NHS algorithm to help us visualize which categories of dots like to be near which other categories of dots. And so it helps us to extract the information that is obvious here, okay, in the color-coded image. But then often biological images are not neatly color-coded, and they have heterogeneous patterns, okay, that are seemingly random. But it is possible to identify distinct patterns of who likes to hang out with who. And this is the end of my presentation. If you are interested in the other algorithms that I've developed, visit my website, tsg-lab.org. And I've developed and am developing automated R scripts to do the calculation for the algorithms, and these scripts are available on my GitHub page here. Thank you very much for your time.